Hello everyone, welcome to the second lecture of our short course. In today's video, we will dive into the fascinating world of genome-wide association studies, or GWAS for short. We will explore what is GWAS, why is it useful, the journey that led us to GWAS, and how GWAS has revolutionized our understanding of the genetic basis of complex traits and diseases. Next, I will talk about the GWAS catalog, a publicly open database for GWAS results, and I will finish with explaining some core concepts in GWAS. So let's get started. GWAS is a method used to investigate genomic variants across the genome to determine their associations with particular traits or diseases. Think of GWAS as a GPS for locating addresses. Imagine you are in a city without maps or GPS, and in order to find a landmark such as 1 o'clock gun at Edinburgh Castle, you might first ask a local where is the Edinburgh Castle. And only when you find and arrive there, you might ask a staff or somebody else where is the 1 o'clock gun. This is because not everyone may be familiar with the 1 o'clock gun, but more people are likely to know about the Edinburgh Castle, where the gun is located. Similarly, GWAS is an initial step to detect vital locations of the genome that are associated with the trait of interest. The goal of GWAS is ambitious yet straightforward. It is to identify genetic variants contributing to traits and disease of interest. So why is GWAS so significant? Why is it useful? It's powerful for several reasons. One, it is hypothesis-free nature. Unlike older approaches that required predefined candidate genes, GWAS scans the entire genome without any prior hypothesis. This is an unbiased approach, and it often leads to the discovery of unexpected but critical genetic variants. And the second point is GWAS pinpoints genetic pathways and mechanisms underlying diseases, opening avenues for drug discovery. And lastly, by identifying associated variants, GWAS improves our ability to predict disease risks, which is a cornerstone of preventive medicine. Now, to understand how GWAS emerged, let's take a look at its historical development, which spans about over a century. Everything begins in the late 19th and early 20th century with two pioneers, Francis Galton and Archibald Gerard. Galton, known as the father of quantitative genetics, he studied the inheritance of traits like height by analyzing the relationship between mid-parent height, which is the average of height of both parents, and child height. Galton introduced the idea of regression to the mean, a key concept in heritability. And around the same time, Gerard described genetic disorders like alkoptonuria, coining the term inborn errors of metabolism. And their work laid the groundwork for linking genes to observable traits. Next major milestone came in 1937 with the work of Haldane and Darwin Bell. They developed the concept of linkage analysis, a method that examines how genetic markers are inherited together within families. And by studying the co-inheritance patterns of genes and traits, linkage analysis allowed researchers to locate genes on chromosomes and identify genetic regions associated with specific traits. This breakthrough provided a foundation for genetic mapping and was instrumental in early efforts to understand the genome's structure and how it functions. And moving forward, in the mid-20th century, we saw many major breakthroughs. In the 1950s and 1960s, genetic screening emerged as a powerful tool. For example, Robert Guthrie developed a simple test for phenylketonuria in 1963, which allows an early detection. And his newborn screening program became mandatory in many places, preventing countless cases of intellectual disability through early dietary management. His test exemplifies how genetic screening can have a profound impact on public health. Next, in 1970s and 1980s, researchers focused on candidate gene studies, where specific genes suspected to influence traits that were tested. This approach was also groundbreaking, but had a significant limitation. It requires prior knowledge or a strong hypothesis about which genes to target. And in 1996, the work of Rich and Americangas shifted attention even more towards the candidate gene association studies, highlighting their potential over linkage analysis for identifying genetic variants associated with complex traits. And after that time, candidate gene association studies becomes even more famous until 2005. The field advanced significantly in 2005 with the first publication of GWAS, which introduced a new paradigm, shifting attention generally from candidate gene association studies to genome-wide hypothesis-free association studies. And unlike candidate gene studies, GWAS systematically scans the entire genome and covering associations without relying on predefined assumptions. And today there are a vast number of GWAS studies, and this actually created a need for a catalog. The GWAS catalog is basically a testament to the success of the GWAS approach, and as of November 2024, it includes over 7,000 publications, nearly 700,000 significant associations, and almost 100,000 full summary statistics. This resource is established by the National Human Genome Research Institute, NHGRI, in 2008, and it reflects the sheer scale of research happening globally. I should also add that there are other online publicly available resources as well, which you can find by simple online research. Let's now cover some key concepts in GWAS. First, genomic variants are a genetic variant is a location in the genome that differs between individuals in a given population. And for a given genetic variant location, we are interested in two questions. One is the variant associated with a trait of interest? If yes, then how much does it impact the trait? For question one, these associations are often visualized in a Manhattan plot, where peaks indicate significant associations. The reason why we see peaks are that, for example, you can see the peaks in here, and the gene names are on top of these peaks. And the reason why we see these peaks are that if a variant is associated with a trait and another variant is highly associated with the original variant, so here we have a variant that is associated with a trait, and we have another variant that is highly correlated with the original variant, then this new variant is also indirectly associated with the trait. Because variants that are close to each other are often highly correlated within each other, most of the times we see peaks as such. And for question two, we define something called effect sizes, which measures the impact of a genetic variant on a particular trait or a phenotype of interest. Okay, and the next concept I would like to explain is linkage disequilibrium, or LD in short. LD refers to the non-random association of alleles at different loci, and it is often visualized in heat maps where triangular patterns indicate blocks of variance that are inherited together. So here we see three LD blocks, and blocks inherited together have high correlation within each other as shown in this heat map. And that's why we see peaks. If one of these are associated with a trait, then most probably all these 5 SNPs are found associated with the trait. And that's why we see those peaks in the Manhattan plots. And the next concept I would like to explain is population structure. And that refers to the fact that differences between populations can confound GWAS results. For instance, oversampling certain populations for cases for example, if you oversample cases in one population and oversample controls in another population, any difference between populations might indicate a false association causing to the disease. And this population structure often measured through plots or metrics such as QQ plot or genomic inflation factor. QQ plot is a plot that on the x-axis we have the expected test statistic, which is often the minus log 10 scale of p-values, and on the y scale we have the observed test statistic, the same test statistics. And what you expect in general in a population without in a GWAS without a population structure is that you see a good alignment for quite number of SNPs, and then for the rest of the SNPs that are found associated with the trait, they deviate from the expected test statistic distribution. And the QQ plot under the scenario of population structure, it starts to deviate from the expected statistic way too early. So you see something like that. And usually you don't only look into QQ plot and say this, but there's also a metric called the genomic inflation factor, which look into the median of observed test statistic and median of the expected test statistic under the null. You can also look at most of the time chi-square test statistic, which is used in GWAS, and median of observed and median of expected. This ratio is called genomic inflation factor, and if it is greater than 1, this might indicate the presence of population structure. Now, in this next slide, I will be talking about the concepts about relationship between individuals. We have two key definitions, identity by state and identity by descent. Identity by state indicates a genomic location that are identical between individuals. And identity by descent indicates genetic segments identical on the same location between individuals, and that is due to inheritance, that are same due to inheritance. For example, if you look into this picture, which is taken from Wikipedia, you see these segments that are identical, and they are both inherited from this individual, so they have the same ancestry. So this is called identity by descent. And using this identity by descent, we define a metric called coefficient of relationship between individuals. And I will not explain the math behind this, but this is basically a measure of biological relationship between individuals. An example is yourself with your child is 0.5 as expected, since you share only roughly half of your genomes with your child. These concepts help measure genetic relationships between individuals. And lastly, I would like to talk about mean rel frequency, Hardy-Vandenberg equilibrium, and genetic relationship matrix. Mean rel frequency is the concept of the frequency of the less common allele in a given population. And Hardy-Vandenberg equilibrium, or in short HWE, describes a population in genetic equilibrium. Both these mean rel frequency and HWE are critical for quality control steps in GWAS. we try to filter out genetic variants that based on these two, especially based on these two metrics, and only after that we run the genetic screening, genome-wide association studies. And lastly, the GRM is the extension of the relationship, coefficient of relationship between individuals, and indeed this is a matrix that estimates the genetic relationship between individuals, and its entries are calculated by this formula. That might look scary for someone who does not have any statistics background, but you don't need to worry about that. The only thing you might need to know is that the expected value of these coefficients, these entries, are coefficient of relationship between individuals. So it is generalizing the concept of our understanding of coefficient of relationship between individuals. And you don't need to know the mathsy details on behind it. But the only thing you need to know is that this is a generalization of the coefficient of relationship between individuals. Okay, this brings us the end of today's video, and we have covered what GWAS is and why is it important. It is historical development and key milestones. We talked about the GWAS catalog, and lastly we talked about some fundamental concepts in GWAS. And in the next video, we will dive into association analysis, linear regression in GWAS, and why we are not using linear regression in GWAS. And then we will discuss about extending it to linear mixed models and the advantages and challenges of using linear mixed models in GWAS. So thank you for watching, and I hope you enjoyed the lecture, and I'll see you on the next video. Bye.